Breast cancer is not a single disease, it's a collection of diseases. It occurs whenever a malignant cell, meaning a bad, harmful tumor, invades the breast cancer cells. And these cells start proliferating aberrantly. So these cells do not function as they normally do. Now this can be localized, meaning it stays in one area. Occasionally it can invade the surrounding area, and this disease can sometimes spread to distant organs. It predominantly affects women. 1 in 9 women are affected, and incidence currently is about 2,800 new cases a year. We have made significant progress in recent years. The survival rates are improving due to the improvement in treatment options, in research, in terms of screening and the awareness programs. However, we're still some way away from transforming breast cancer from being a fatal disease into a more manageable chronic long-term disease. Breast Cancer Ireland supports a number of research centres nationally. In our particular lab, we focus on the hormone receptor positive breast cancer. So these are breast cancers that use hormones such as estrogen as fuel, which make the cells grow and spread. The current conventional therapies for hormone receptor positive breast cancer are endocrine therapies, and these work by blocking the hormone receptor and preventing this fuel from getting into the cell, thereby preventing the tumor cells from growing and from spreading. So we use a number of different molecular techniques, a number of different tools, most important being the breast cancer cells themselves and the patient tissues. So Breast Cancer Ireland sponsors a number of specialist nurses nationally in various different Breast Cancer Ireland centres. And all these research nurses recruit patients onto our studies, and they help us collect tissues and bring them all together in one centralized biobank, which we then are able to use for our research purposes. This offers us an opportunity to have a unique translational research where we're able to take our lab research and translate it into future treatments that will be translated back to the patient. In our group, we tend to focus on oncogenes. So these are genes that are normally characterized as being very important in cancer. And these are faulty genes, genes that send a relentless signal back to cancer cells to tell them to proliferate. And what we can do is we can delete these genes from our cells. We can put them back into the cells. What we can also do is then we can see if there's some different way for us to manipulate it and see if we can switch them off. By doing so, we can see what other components are important for tumor progression. We can have a look at these cells and see whether they're important in terms of tumor invasion, in terms of tumor progression and tumor metastasis. When we have found something interesting in the lab and something that we think that is important in terms of cancer progression, we can really use our collections and our connection with the research nurses, and we can utilize our patient samples to really link what we do in the lab and really connect the breast cancer cells, the models that we use in the lab, with those of patients. And by doing so, we can collate all this data together and use it to predict which patients will do well on therapy, which patients might relapse on therapy, and to help us better understand how we can actually overcome and help the significant portion of patients that don't ultimately benefit. The ultimate goal for us would be to find a biomarker that we could potentially test out in a standard blood test, which will be able to tell you whether the patient can respond to the therapy. If they're a responder, great, give them the therapy. But if this biomarker is high in the blood, we should be able to say this patient might actually relapse on therapy after a number of years. And this is ultimately leading to a more personalized approach to medicine and not doing a one-for-all treatment. One of the studies that we're currently doing involves advanced disease, and we've looked at patients that actually have disease that is spread to distant organs. And for example, it's really fascinating and scary to think that a breast cancer cell can invade, travel in the blood, survive, and then get into the brain, survive in the brain, hide for years, thrive, and then all of a sudden you get a breast tumor growing in the brain. And what we're doing is we're tracking the changes genetically on the DNA level, the changes that occur over the years to the primary tumor. So how has the primary tumor changed to allow it to go to the brain, and survive in the brain. And the way we do this is we sequence our patients, so we do sequencing on the DNA and the genetic level, and then we use specialist bioinformatics software to tell us what exactly are those changes and which are the faulty genes, which we can then target using our molecular techniques in the lab, and we can potentially find better suitable therapies that will prevent this from happening. So what we do is we tend to take our cells and we tag them with this luciferase. So it's a firefly gene, so a gene that's found in fireflies that makes the fireflies glow. And we put that into our cells and we tag our cells. So whenever we do anything to our cells, if we look at them under the microscope, we can track their progress because they glow either green or they can glow maybe even red, depending on what you choose. And what we can do is we can alter one of those genes, change it, delete it, put it back in, and see if we can prevent the cells from invading, and we can see if we can prevent the cells from migrating, and we can also see if we can give it an additional treatment or something different that will actually stop these cells from growing, and it will stop them from actually surviving. In one of the other labs, there was a recent study, a very high-profile study. They were able to find that a combination treatment of two different tyrosine kinase inhibitors was actually better in terms of survival for a specific subset or a specific cohort of patients. Studies such as these that involve huge international collaborations and clinical trials have recently brought to light that this new combination was very successful. About 50% of patients, it was able to get a complete clinical response where it completely eradicated the tumor. And this really kind of brings us forward to getting to a stage where we can focus on specific cohorts of patients where we don't need to give chemotherapy, and we can spare them the side effects of chemotherapy, and potentially we can, whenever these are given as neoadjuvants, meaning they're given before the surgery ever even takes place, if we can find the patients where the tumors are completely eradicated, we might not even have to do surgery, and it will spare complications from surgery, and you will get to a stage where you can give these cocktails of very specific drugs depending on your tumor profile. On the genetic level, on the DNA level, every tumor has its own profile. And what we can do in the lab is we can use the resources that we have to sequence these tumors, look at the profile, so look at all the genes and how they're expressed and how they're different to the other ones. We can see what's the best sort of solution in terms of treatment for the patient, and then we can relay this information back to our clinicians and oncologists that should be able to determine the best treatment course. This is the sort of research that Breast Cancer Ireland funds and supports.